So today we would begin with this topic, biotechnology, principles and processes. And you must have seen in the NCERT the definition given for biotechnology from European Federation of Biotechnology. So according to them, it is because you see, whatever we are doing in biotechnology, that exists in nature already, but we are manipulating it according to our own benefit. So this entire biotechnology, it involves two basic techniques, or two core techniques are there in this. Two core techniques. One is genetic engineering, where of course we alter the DNA of an organism, and so this involves creation of recombinant DNA, where we can combine DNA of two different species, and that would be a recombinant. And this is not to be confused with the recombination that we study in the process of meiosis, because that is something which is happening naturally. Here this recombinant DNA, like you all have studied the preparation of humulin, that is human insulin gene is attached to the plasmid of a E. coli. Now such type of new DNA which is created, this is not existing in nature, rather it has been created in the lab. So that is the recombinant DNA. Okay, so creation of recombinant DNA, then this will be transferred into another organisms or the host cell, so that is gene transfer, and also making copies of it, that would be gene cloning. So this step involves all of it. Then another one is the bioprocess engineering. Now bioprocess engineering is maintaining a ambient environment, because bacteria, like in replica plating experiment, I told you that we will be making a agar plate with all the nutrients on it, and on that we can grow the bacterial culture. But at the same time we have to maintain it so that other fungal spores are there, other bacteria are there in the atmosphere, they do not reach to our culture medium and grow along with the desired microbe. So only the desired microbe should be growing there. So that's what is the maintaining an ambient culture, maintenance of ambient culture medium, and this we are using along with antibiotics, because our desired microbe is resistant to antibiotics while the others they are not, so they would die on that medium. Okay, so maintenance of ambient culture medium having or using antibiotics, allowing the growth of only desired microbes. Okay, so these are the basic techniques. Now coming to the first one, that is where we have to change the DNA, the entire DNA which is having number of genes in it. So when we want to manipulate the DNA, we would be requiring certain tools. So the entire process of genetic engineering, you see, it requires tools. So tools for a genetic engineer, for genetic engineering we can just write in short as JEE. So tools for JEE, one is enzymes, then other is the vectors which will be required for gene transfer, and of course in which this gene will be multiplying, those are the hosts. So we require these three things, you see. And so first one, starting with enzymes. Now The first and foremost, the most important enzyme required in genetic engineering is where you can cut the DNA at a specific sites. So that one is the restriction endonucleases. Restriction endonucleases, or simply in short we can write them as RE. Now these restriction endonucleases, first thing, they are found only in prokaryotes, that is in bacteria. Few days back there was a question in the NTA app where in the viruses they had mentioned, but that was a wrong one, where it should have been a restriction or reverse transcriptase, and maybe that was to be typed and restriction endonuclease was typed. Don't worry about that, because these are found in bacteria and bacteria only. Okay, so these restriction endonucleases, these are found in bacteria. And why they have been called restriction? Because this is to restrict the growth of the bacteriophage, the viruses which infect the bacteria, those are the bacteriophage, and when that bacteriophage puts its DNA into the bacterial cell, the bacteria who is producing this restriction endonuclease will cut that viral DNA and the virus won't be able to multiply inside the host cell. Okay, so found in bacteria as a defense mechanism, against invading viruses. If it is written viruses or bacteriophage, any one of it will do. Okay. Now the other thing is, if we talk about these nucleases, because endonucleases, that belongs to a group of enzymes called nucleases which cut the DNA. Okay, so let us mention this, nucleases, and these nucleases are enzymes that cleave or hydrolyze, you can say, that hydrolyze term is given, cleave, cut, anything, doesn't matter, because basically these are hydrolases. So with addition of water, it is going to cut what? The phosphodiester bond. So enzymes that cleave phosphodiester bonds, right? Now these nucleases, they are of two types. One is exonucleases and the other is endonucleases. So in digestion, if you remember, protein digesting enzymes, the proteases also I had classified as endopeptidases and exopeptidases, same terminology here also. So which will cleave a terminal peptide phosphodiester bond, so that would be called a exonuclease. So suppose here we have a DNA, and in this DNA the terminal most bond is cleaved. So in that case the chain gets shortened, and this would be a work of exonuclease. So these cleave terminal phosphodiester bonds. Okay, and endonucleases, these cleave interior bond, a interior phosphodiester bond. So a long fragment of DNA is cut into smaller fragments. So these cleave interior phosphodiester bond, and smaller fragments from large one will be developing, are formed. Okay, so here you can see the restriction endonucleases, they belong to this group. Now in this restriction endonucleases also till date have been different types have been formed. Now this NCERT, the NCERT that you have with you, this came out in I think 2005. At that time it was of course 900 restriction endonucleases were discovered, but now that number has increased so much. It is more than 2500 or so, but anyway, it doesn't matter. We'll follow that line, that nearly 900 restriction endonucleases from various bacteria have been discovered. Okay, now these restriction endonucleases, what is special about them? So in that, now before I go further into restriction endonucleases, one more enzyme let me mention. Now you see, when the bacteria is producing restriction endonuclease, its own DNA is protected from this restriction endonuclease by an enzyme that is called methylase. Okay, so methylases protect bacterial DNA from being digested. It's anyway called restriction digestion, because all hydrolytic enzymes are digestion, only they are carrying out, from being digested by RE. Okay, protects the bacterial DNA from being digested by RE. How does it do? Because just now we'll be mentioning that restriction endonuclease read a specific site on the DNA. There is a specific site on which it will be acting. The same site is identified by methylase also, but wherever there is a cytosine, it will put a methyl group there. So it is basically, you see the structure part, the shape will be changing, and enzyme cannot act over there. Okay, so this one we can just mention, it adds methyl group to cytosines, and that way it is protected. Now coming back to this restriction endonucleases. Now restriction endonucleases, they are very specific. Okay, and without going into much detail, let me just mention one thing, that restriction endonucleases type 1, type 2, type 3 are there. Type 2 are the most specific, and for genetic engineering, type 2 restriction endonucleases are used. Okay, so coming to this, type 2 restriction endonucleases are most specific out of the other restriction endonucleases, most specific, and cut at a specific site, and this site is a palindromic site, which is palindromic, usually 4 to 8 nucleotides long, so it will be reading that site, and this site of course will be called accordingly the restriction site or the recognition site. Now the first restriction endonuclease to be discovered, that was by Arbor, Smith and Nathans. So Arbor, Smith and Nathans discovered first restriction enzyme, and this one was Hindi2. This is how it is pronounced, not Hind, okay, Hindi2, where the source is, you see, this is the Haemophilus influenzae, that is the name of the bacteria. And if you remember, this is the bacteria which causes pneumonia in Human Health and Disease. We have mentioned the name of this bacteria. So you see, from the genus name, the first letter is taken. From the species epithet, we take the 2 letters. So that is Hin here. And D, this comes from the strain. Now bacteria have different strains. So the strain is RD. So from here, this D is taken. And this is, now 2 would be the order of discovery. Okay, so order of discovery, and this is usually the common query, that if it is 2, then how come it is first? So let me tell you, all bacterial enzymes, it is not only restriction endonucleases, all bacterial enzymes are named in the same manner. The naming mechanism or the naming protocol is same. Like you see TAC polymerase. So TAC polymerase, TAQ, that is Thermus aquaticus, but that one is a DNA polymerase. So all bacterial enzymes are named in the same manner. So from this Haemophilus, the first enzyme to be discovered might have been something else, maybe oxidoreductase or lyase, I don't know. But the second one was this, and this one among restriction endonucleases is first. So it is first among restriction endonucleases, but second from this bacteria. Okay, so that is the one, and its restriction site or recognition site, that is 5' GT and here 3'. Now PY is pyrimidine, it could be any pyrimidine, and here purine, it could be any purine, and here it will be then accordingly CA and TG. So you can see it is palindromic, that if you read from here, then in the other one it is the same sequence, but if it is read from the opposite side. Okay, now where does it cut? It cuts from here between the purine and the pyrimidine. Here it cuts, so that the two ends which are going to be created, these are PY and this is CAPU. So this you see, this is the one, and same way the other one would be PU, PY, and this is ACTG, like this. So such ends, if these are created, these are called blunt ends or flush ends, where these are equal. Now these are not useful for genetic engineering until unless we can change them and make them sticky ends, but usually we will try first for another restriction endonuclease which provides us with a sticky ends. Okay, so these are not so useful. Then coming to another example where we can see sticky ends being produced. So another example we can take of restriction endonuclease, that is ECO-R1. Then in this, once again you see the source is the Escherichia coli. So taking E from here, CO from here. The strain is RY13, so R from here, and first is of course the order of discovery. So here the restriction site is restriction site. This is 5' GAA TTC 3', and here this would be C TT AA G 5'. Now this one is cut by here between G and A. Between G and A, here it cuts on both these strands. So now the here G and here C TT AA, here it is AA TT C, and this is G 5'. So here you see there are 4 nucleotides which are unpaired. They are hanging. This is called an overhang. And here also you see the 4 nucleotides, these are hanging. And this is what is called sticky ends. So sticky or cohesive or staggered ends. These are created. Okay. Now this is what we need, because in this case what has to be done, that the two different DNA, that is, let us say one is bacterial DNA and the other is eukaryotic DNA. So both the DNA which we want to attach them together, we will cut them by using a restriction endonuclease which gives us a sticky end, and the same restriction endonuclease we will use for both, that is the vector DNA as well as the DNA of interest, and as a result same type of sticky ends are produced, and then it is easier to put them together, right? So here we can just mention that both vector DNA and gene of interest or DNA of interest and DNA of interest are cut with same restriction endonuclease to get same type of sticky ends. Now you see, previously there was one question where 4 restriction endonucleases were given and you had to identify that which one gives a blunt end. So you see, as I told you, there are thousands and thousands of restriction endonucleases discovered so far. So it is not easy that you can remember all of them, but certainly we can take few examples, like 2 examples we have already done. So restriction endonuclease that produce blunt ends. That year when it had come, it was ECO R5. ECO R5 was there. Then another one we can mention is SMA1. SMA1, this produces a blunt end. And then that produce sticky ends. This one is easy because more are there with this, like this, some of them we will be doing anyways. BAM H1, PVU1, PST1, SAL1, EcoR1, this all we have already done, right? So we can, and that year there was one option was XHO1, that is from Xanthomonas holicola, even that one is producing sticky and so on. So from here then we can move to the next. Now we have cut our DNA. So next is molecular pasting. So this was molecular scissors at their work. Now coming to molecular pasting. So second category of enzyme we are going to put is ligases. Ligases. Now you see, when we bring this together, like here itself, let me put it here, like I'll just remove one side, and suppose we have a DNA from another source, that's why I'm putting a different color. They have been cut, both of them by EcoR1, and in this you will see there would be AATT, and here it is C, here it is G, 3 prime, 5 prime. So when you bring them together, the hydrogen bonds, they are formed spontaneously, there is no problem there. Ligases, why do we need? Because these hydrogen bonds, you know, they are weak bonds, they can form and they can detach easily if slight heating is there, but they are sealed. They are sealed, these two fragments are sealed because there has to be formation of a phosphodiester bond here and a phosphodiester bond here. So when the phosphodiester bonds are formed, this will be then permanently attached, and that bond is formed by ligase. So ligases, you see, these are molecular paste, that's why it is called, and these bind 2 fragments of DNA, 2 fragments of DNA by catalyzing formation of phosphodiester bonds. Okay, and which ligase do we use? That has been taken from a virus, that is T4 ligase. T4 ligase is used in genetic engineering. Okay, because viruses, they do have this. They can cut bacterial DNA and then attach their own DNA there. So that they do need endonucleases undoubtedly, but that endonuclease won't be for at a specific site. So it shouldn't be restriction endonuclease. It could be a general endonuclease. Okay. So anyway, moving on to next. Now suppose we didn't have a choice because you have different types of scissors with you, but the thing that you need to cut, you have to use a specific scissor. And so the restriction endonuclease, like suppose your DNA of interest didn't have a site like this, that we could cut it by EcoR1 or by any other restriction endonuclease, their restriction site, that was not there. We have to cut it in this manner only. So we are getting a blunt end. So these blunt ends we can convert into sticky ends. So for that, what we use is another enzyme, the third enzyme next we can talk about. I'll keep this so that you know what has to be put. The third category of enzyme, that is terminal transferase. Terminal transferase. So this one is used to convert blunt ends into sticky ends. Okay. So what does it do? Terminal transferase. Now you see the name itself is transferase. So it is transferring a nucleotide at the terminal end, where that would be at the 3' end. So here you see this is 5', this side would be 3' end. So here 3' end, it would be same type of homonucleotide. If it is transferring adenine, so adenine every time. So adenine, adenine, adenine. So now you see a overhang is created. And because of that, now it has become a sticky part. Same way on the other DNA which we want to attach with it, we will create a thymine overhang there, and now these two fragments can easily bind together. So this one transfers homonucleotide at 3', at the 3' end, you know there would be a OH free, so we can write it like this also, 3' OH end, and thus a overhang is created. So that is one, terminal transferase. The name itself are terminal transferases only. And then another one, now when we have a DNA of interest, we want to make copies of it. So for that we require DNA polymerase. So this is used to make copies of desired DNA. Now this part you have already done in botany. So you have studied that in prokaryotes also you have studied there are 3 of them, that is DNA Pol 1, Pol 2, Pol 3 are there. Out of that, Pol 3 is very fast, but Pol 1 is having a exonuclease activity also, because it keeps on proofreading also. So as soon as a wrong nucleotide is attached, it will go in the reverse order, remove that nucleotide so that the correct nucleotide can be attached. So here we want speed, but at the same time we want accuracy as well. And that goes for you also. When you are solving the question paper, you have to have a speed, because in 3 hours, 180 minutes, 180 questions, but at the same time accuracy is also important, because every wrong question gives you -1. So all the hard work is gone, right? So here DNA Pol 1 is the enzyme of choice. DNA Pol 1 is enzyme of choice, as it is more accurate. More accurate. And in PCR, you know that we use Taq polymerase, that is from Thermus aquaticus. Taq polymerase is used, and this Taq polymerase is actually in the category of DNA Pol 1 only. Okay, so this is actually DNA Pol 1. Right, this is one. And last one more enzyme I would mention, and then we are through with the enzymes. That is alkaline phosphatase. Alkaline phosphatase. Now you see here such huge we have drawn, but normally what happens is, I'll just remove this side. That suppose here you had the plasmid, and on this plasmid you used a certain restriction endonuclease, so that this plasmid is cut, and here we have two sticky ends formed, right, in this manner. And there is our DNA of interest. Now this DNA of interest is also cut with the same restriction endonuclease, so that here also the same sticky ends are produced. Okay, so this is the DNA of interest or gene of interest. Now we put both of these together. Yes, let me put a different color. And this is nothing is visible by naked eye, you see. We put both of these together and we put our ligase, and we want that this attaches here, and so that a recombinant DNA is formed. But quite often what happens is that these two ends, these are sticky ends, they will bind together, and that's what is called self-ligation. Now to prevent this self-ligation from occurring, what is done, that you know that between two nucleotides where phosphodiester bond is formed, there is only one phosphate. So if that phosphate is removed, then in that case the self-ligation would be prevented. While the other one still has the phosphate at the end, so no problem, that phosphate will be used. So to prevent that, we use alkaline phosphatase. But at the same time, very small amount has to be put, because otherwise it will remove more and more other phosphates also. So this one removes a phosphate at the 5' end, and that will prevent self-ligation. Okay, so this one removes phosphate from 5' end, and this prevents self-ligation. Okay, that is there. And in this usually bovine alkaline phosphatase is used, or calf also we can write. This is how in short form we'll write bovine is, which has been taken from the cattle. So bovine alkaline phosphatase, there is AP, of course you can make that this is alkaline phosphatase. So these are the different enzymes which a genetic engineer would require. Now coming to the next thing, that is the vector. Now the vector, just like your NCERT gives the example, like for malaria, mosquito is the vector which is taking the plasmodium from into the body of the human. So same way here, because this DNA, it cannot undergo replication, or it cannot express itself in a external medium. It requires a cell in which the replication will occur. Gene cloning can only take place then. So there has to be, and this small fragment of DNA, this also doesn't have the ability to replicate. So it has to be part of an entire chromosome or entire DNA. So that DNA which will take the gene of interest, or the gene of the DNA of interest into the body of the host, that will be called vector DNA. Okay. So next thing we can first mention, because here you see plasmid one is already drawn, this is the vector DNA. So you see this is the vector DNA, and this is the DNA of interest. DNA of interest, or foreign DNA, alien DNA, any such term you can use. And once this attaches, suppose here it does attach, then what is formed is recombinant DNA, right? Once it attaches here, this is recombinant DNA. Okay, so starting with this vector DNA, I am not using the word cloning vector as yet, I'll tell you, because even that has been a question. We'll come to that although. Vector DNA. So vector DNA is a DNA. Okay, so it is a DNA which transfers gene of interest into the host cell, where it can make its copy, where it can replicate, that is, it can express, okay, where it can clone, express, okay. So that is what is vector DNA. Now here taking up a plasmid, which is used as a vector DNA, because there are so many of them which are used. So first of all, starting with plasmid. Now plasmid, you see, is extra chromosomal double stranded DNA found in some bacteria, not all of course, okay. So this is extra chromosomal autonomously replicating, because it has its own replication sequence, okay, autonomously replicating double stranded DNA, double stranded circular, we can also add on the word naked, because you know that prokaryotic DNA is without histones, naked DNA found in, once again, bacteria, okay. Now as in which form it is found in the bacteria, the same is not being used, it has been modified so that it can be used as a vector, okay. And this work was initially done by Stanley Cohen. So you will see in the biotechnology where the unit starts, you will find Herbert Boyer, the write-up is there with a photograph, and Herbert Boyer and Stanley Cohen, they were working together. Herbert Boyer was using these endonucleases, restriction endonucleases, the use of it he had learned, while plasmids Stanley Cohen had extracted and he was able to modify them. So both of them, they combined their work, and that is how they started creating the recombinant DNA, okay. So this vector DNA, you see, it is used by different, it can be given different names, like this vector DNA can also be called passenger DNA, passenger would be that, this is vehicle DNA, vehicle DNA, or gene taxi, any such, so many different terms can be used on it. And who will be sitting on this vehicle? That is of course the DNA of interest. So this DNA of interest can be called passenger DNA. This is the passenger DNA. And when both of these combine, so this is of course the recombinant DNA, but this recombinant DNA can also be called chimeric DNA. Chimera, you see, you have studied fish, cartilage fish, which has bony fish characteristics also, but otherwise chimera is in the Greek mythology, it is a monster who has the head of a lion, body of a goat and tail of a serpent. So anything which is a mixture, we in scientific jargon also it is called chimera. So a chimeric DNA, where the DNA from two different species, two different sources has been joined together, so that has been also named chimeric DNA. Okay, So anyway, coming back to this, so this plasmid, so number of plasmids have been developed. So Stanley Cohen, he developed plasmids. Yeah, the example like it is given that from one of the bacteria, he took out the antibiotic resistant gene, put it in the plasmid, and then he transferred that plasmid in E. coli. Now E. coli also developed resistance against antibiotics. So from one bacterial species to another bacterial species, Salmonella typhimurium to E. coli. So his plasmids, he named them as PSC like this, PSC 101, PSC 102 like that. But we are not going to that is just part of the history. We'll rather talk about the plasmid which are used in the various genetic engineering experiments. So we'll take PBr 322. Okay, so PBr 322. Now this, how the naming is. So once again, P is for plasmid. This P stands for plasmid. BR is the researchers who developed it, that is Bolivar and Rodriguez. And 322 is the number. So they must have prepared number of plasmids, and in which the successful plasmid which could be actually utilized, that is 322. And there is even PBr 327, 330, so on, number of them are there, but we are of course talking about 322. Okay, so that is the experiment number, or to differentiate it from other plasmids that they created. Now this plasmid, we'll mark it here. Now yes, so we'll take each part of it one by one. So this is ORI, ROP, then here there is a gene. Of course, in actual plasmid these are not colored or anything. This is for our own understanding, for convenience we are doing it. And similarly here there is another one. So this is Ampicillin resistance gene. This one is Tetracycline resistance gene. That is, now since you have studied genetics, so you know that a gene is responsible for making a particular polypeptide. Now this polypeptide which is formed, this is an enzyme which will break down ampicillin. So in the ampicillin medium, the bacteria which is having this gene will be able to survive. Same way, from this tetracycline resistant gene, there is an enzyme being synthesized, which enzyme can break down tetracycline. So a bacteria which is having both the genes intact will be able to survive in both the antibiotic medium, right? Then there are number of restriction sites which are shown in your NCERT, but only two you need to remember, or two here. That is, one is BAM-H1, another one is SAL1. So if we use BAM-H1, it is going to cut from here, or SAL1, that means through the tetracycline resistant gene, that's it. And here we have PVU1, PST1, PST1, like this. Okay. So these many will do. And these all four which are mentioned, these are all producing sticky ends. So now what from here we'll take on the properties. What properties does should a vector have? Okay, a vector, a DNA to be used as a vector, it should have certain suitable properties. So in them, one is of course that one we have already mentioned, like it should be able to autonomously replicate, so that way it will be easy. And then it should be small, because DNA is a very large molecule on its own, and to cross that DNA through the cell boundary into the cell and taking out of the cell, that is very difficult. So it has to be small sized. Okay, So these are already there. Then rest of the things we can mention, that is just a sec. Coming on to first thing, that is ORI. ORI is origin of replication. Origin of replication. So origin of replication is where when this DNA is to be copied, that its copies are going to be formed. So for that, the gene which is regulating, which is initiating the copying, that is from the ORI. So replication, okay. So origin of replication. So this one is responsible for initiating replication, and how many times it should replicate, and maintaining copy number. Because normally what happens, whenever you see a diagram of a bacteria, in that the bacterial chromosome, that is the genophore or the nucleoid, that is a highly coiled, a single DNA there, which is once again circular double stranded, but only one copy. And in this extra chromosomal plasmid is just shown one like that, usual diagrams. But this one can make copies, so that there will be number of copies of this. Okay. So a plasmid in which high copy number can be maintained, those are preferred, of course, because you see, if we have put a gene of interest here, so as many copies this plasmid is going to form, that many copies of the gene of interest also are there. So gene gets cloned in that manner, right? So there are plasmids where they make 500 copies of themselves. So that means they are making 500 copies. So our gene of interest also is copied 500 times. And when this bacteria divides, then further they are passed on to the next generation of bacteria also. So this is what is the function where which type we need. Okay. So we can just mention high copy number is preferred. So this is one. Then ROP here, which is mentioned, that is replication of plasmid, in which there are, this part will be coding for certain enzymes, proteins, which will be needed at the time of the replication to take place. So this is there in the larger plasmids. This one is quite large, 4.3 kb it is, but in the smaller plasmids, ROP is not there. They will utilize the proteins which are already there in the bacterial cytoplasm. Okay, but in this it is present. So ROP is, that stands for replication of plasmid. So this one codes for proteins needed for replication. Okay, but this is not a necessary item. This is, as I said, in smaller plasmids, ROP is not there. ORI should be there. So this is absolutely necessary. Okay, so you will see in the NCERT even there is a restriction site given on ORI. So if that restriction site we use and from here it is cut and gene of interest gets incorporated here, then it's useless because now the ORI cannot work. So we won't be able to make copies of it. So ORI has to remain intact. Then coming to this antibiotic resistant genes that are there, these are the selectable markers. Okay, so next very important thing is selectable markers. Now selectable markers, why are they needed? These are needed because here we have drawn such a big thing. This is all nothing visible to us. So these are needed to find out whether our vector DNA along or the recombinant DNA has been able to enter into the host cell or not, and even if it has entered, whether it is carrying the gene of interest with it or not. So two things we need to know. Okay, so these are first thing, these are needed to find whether vector DNA has entered into the host, and any DNA from outside, if it is entering into the host, so that process will be called transformation, and that host will be called a transformant. Okay, so we want to identify the transformants from the non-transformants, and whether the vector DNA has gene of interest in it, so those transformants will be called recombinants. Okay, so those transformants who have taken the vector DNA and they have taken the correct vector DNA along with gene of interest was also integrated. Okay, so those transformants are called recombinants. So you see, that is the reason we are requiring two selectable markers. One selectable marker to detect transformants from non-transformants. Then among transformants, we have recombinants and non-recombinants. So we have to select at two levels. So screening has to be done at two levels, right? And so here mostly one of them is, later I'll be mentioning another one, PUC8. This one is using two different antibiotic resistance. So in this way, this will be better understood if we take an example. Like suppose if we use BAM-H1. Okay, so example, use BAM-H1. Now when we use BAM-H1, so that means it is going to cut somewhere here, and when it cuts here, I've just made a straight line, of course it's a sticky end, and then we use our gene of interest, and suppose this gene of interest properly gets integrated. So use gene of interest and then ligate with vector DNA, with the gene of interest or a foreign DNA with foreign DNA. So if it does get ligated, then what is going to happen? That here let us suppose here our gene of interest has come. This is the place where gene of interest is there, and the tetracycline resistance gene is one part here, now one part here. So it has been now split. We haven't touched the antibiotic, the ampicillin resistant gene. It is as such, no problem there. It is as such, and of course ORI and ROP, no effect there. Okay. Now, so here this is the foreign DNA part. Foreign DNA part. Now this would be called the recombinant plasmid. Recombinant plasmid. While this one, because even though we use alkaline phosphatase, but still, because out of so many plasmid molecules we are having, some of them will certainly self-ligate. So one of it, let us say, remains self-ligates, and so it will be just the original form. So this one will be the non-recombinant plasmid. Now when you take these plasmids, so in your sample now you have recombinant plasmids as well as non-recombinant plasmids. You don't know about it. You just carry out the process of transformation of E. coli. So out of that E. coli, then we'll have to screen. So screening, next step after So after transformation step, after transformation, there is requirement of screening, requirement of screening. Okay, so this is the important step. So in this case, what is done is that I'll just move, this is not needed, yes. So now you see we have three types of bacteria with us. One in which the plasmid couldn't get in at all. Okay, so let us put it in the form of a diagram, that way it will be easy. So here, no plasmid. Here plasmid did enter, but it is non-recombinant. And third one in which the plasmid entered and this plasmid is also having the gene of interest with it. I hope everybody can see this, right? Yes, it is visible. So this one is non-transformant. This one is non-transformant, in which the plasmid didn't go at all. And these two are transformants. Okay, so these two are transformants. But in these two transformants, this one is, you see here the gene of interest didn't get in. So this is non-recombinant. And this one is recombinant. So I hope up till here it should be clear. Now the thing is, how will we screen them out? So first thing that we do is that we prepare a plate. Okay, I am just putting the plate from the top so that you are seeing the petri dish from the top. And on this petri dish we have our agar and complete medium, everything. So this is agar medium. I am just writing agar medium now since I have done with you the replica plating. So you know that it is agar and the nutrients are there. And in this but you have also added what? Ampicillin. Ampicillin. Okay. So now on this plate, who will be able to grow? These cannot grow because they don't have the antibiotic resistant gene, ampicillin resistant gene. But both of these will grow because both of them have antibiotic resistance. Okay, so that means we get colonies here. And these would be colonies. We'll have to incubate. We transfer the bacteria and then we incubate it. And then after that, we get a plate on which we have these colonies. So these are transformant colonies. Transformant colonies. Okay. Now you prepare another plate. And in this plate, what you do is you take agar medium, once again, same way. But with this you add tetracycline. Okay. So just like in case of replica plating experiment, I had told you that we have a wooden block over which we have placed a velvet cloth. We press here, some bacteria from each colony they stick, and then we press here. And when we press here, what do we find? That some colonies, I am just putting this mark, that is just to show कि here there should have been colony but it is not there, and only few colonies develop. Okay, these are empty spaces, right? So who will be able to develop? The one in which the tetracycline gene is working. In this case, the tetracycline gene will not work because that gene has been cut and in between there is a stretch of DNA which is not part of that gene. So the gene has to be in continuation, then only it can work. So in this case, you see what has happened? The tetracycline gene has been insertionally inactivated, right? So tetracycline resistant gene is insertionally inactivated. Okay, so where tetracycline resistant gene is not working, those colonies will not be able to develop here. So the colonies which will develop, those will be whom? The non-recombinant, because in non-recombinants the tetracycline gene is intact. So these are non-recombinant colonies. This is non-recombinant colony, these stuff. And these empty spaces should have been what? The recombinant colonies, but you have killed them. Recombinant colonies. So that is the reason why replica plating, why is to be done, because on this plate recombinant colonies you will not get. But since it is an exact copy of this master plate, we know now which colonies are there which are having recombinants in them. So we can pick the bacteria from there now. Okay, so that's why the screening is very difficult here in case when we are using PBr 322, because we have to prepare two plates. One plate first with ampicillin, by which we are able to select transformants, non-transformants die. Then out of these transformants we have to select recombinants and non-recombinants. I think it should be clear up till here, right? So this is, that's why you see our NCERT also writes that it's a cumbersome process because it requires plate, they are making two plates, and there is always a danger that by mistake on this plate if you put tetracycline, that means the whole work is lost because the recombinants would die, right? So that is why to get rid of this problem, other plasmids were prepared. And one of them which I am going to mention with you is PUC8. Okay. So next one. PUC8. This one was prepared in the research lab of University of California. So UC is University of California. Now in this plasmid, this is a comparatively smaller plasmid. And in this, one is same, that is ampicillin resistant gene is still there. Okay, but instead of tetracycline resistant gene, what has been taken is LAC-Z, and ORI is there. This is ORI, this is ampicillin resistant gene, and this one here is LAC-Z, LAC-Z, whichever way you can call it. And within this LAC-Z there is a region, okay, here there is a region which is called MCS. MCS, that is multiple cloning site, multiple cloning site or polylinker. Now within this small site there are number of restriction sites, huge number of restriction sites. Like just for example I am giving you, like within this there is ECO R1, BAM H1, Hindi 2, SAL 1, so on. Number of them are there. Okay, so we have a choice that which restriction site we want to use, but we will be always putting our gene of interest at the MCS only. Okay, and when if it is properly integrated, ligated properly at the MCS, that means the LAC-Z gene gets insertionally inactivated. Okay, so in this case, after successful ligation, the LAC-Z gene is insertionally inactivated. So this gene will not work. Now here the screening becomes easy. So in this screening, if we talk about, it's called blue-white screening. Blue-white screening. This NCERT does mention, although the details and all they don't give. Okay. So in this case, what is done? That once again we put a plate, take a plate, prepare it with agar medium and ampicillin. Okay. So here we have the plate, and on this plate we have taken agar medium plus ampicillin. So now you know only what will grow on this medium, only the transformant colonies will grow. So here transformant colonies, they develop. Okay. And then what we do is that this gene LAC-Z, this one codes for an enzyme beta galactosidase. So this also you must have studied in the lac-operon model. So beta galactosidase enzyme, which breaks down lactose to glucose and galactose, right? So here the substrate is being put. Now substrate not directly galactose, but it has been modified, but the enzyme can still act, that is called X-gal. So here not what is done is that we put X-gal. X-gal here can be called chromogenic substrate. Chromogenic substrate. Why chromogenic? Because when this substrate is acted over by the enzyme beta galactosidase, the product which is formed is blue in color. So what happens, because otherwise if it is just lactose, the product which is formed, galactose and glucose, they are also white, and lactose is also white, you know that. So here what is going to happen is that some colonies, they will be white in color, they will remain white, and it's very clearly with naked eye it is visible. Okay, some are white while some turn blue. So that's why we are using the term blue-white screening. Some turn blue. Now the ones which have turned blue, that means, just a sec, I'll first write here. So you see this X-gal, which itself is white in color, and this by the enzyme beta galactosidase forms a blue product. Okay. So the ones which are forming a blue colored product, that means the enzyme is working, and if the enzyme is working, that means the LAC-Z is working, and LAC-Z is working, that means it is non-recombinant. Okay, so this is non-recombinant colony. So non-recombinant colony would be blue, and the white colony where X-gal remains white, it is not being broken up, that means the enzyme is not being formed. So this white colony is of recombinant. Recombinant colony. This is white, right? So by this one single plate we require, on one single plate we are able to screen, and it is comparatively easier, and by mistake you are not going to kill the recombinants. So this makes the, because here whenever some type of experiment is done where we need it in the lab to do again and again, we have to simplify things, and this has made the selection process much simpler, right? Now lastly we can just mention one thing, that is how much insert load can they carry. Okay, insert load is, that is the vector DNA is having certain amount of DNA, and on this how much foreign DNA can we add to it, because since we mentioned it, it's a gene taxi or a vehicle DNA. So every vehicle has its limit, how much load it can carry. So here same way we are using the term insert load. Okay, so insert load that can be transferred by plasmid vector, that can be transferred by plasmid vectors is within 5 to 10 kb, not more than that. 5 to 10 kb, that would be, kb is kilo base pairs, while the plasmid itself, like PBr 322, is itself 4.3 kb. So of course it is carrying more than that. This one is smaller, about 2.3 kb, PF8. So accordingly that's how the range is, okay, but less than 10 kb, within 10 kb, not more than that. So this is one vector. Next vector we can talk about is bacteriophage. Now bacteriophage, as we said, these are viruses which infect the bacteria, and they have this natural ability that they can enter the bacteria and integrate their own DNA with the bacterial DNA. And then of course, but they will be killing the bacteria. So that we don't want. So we have to modify the bacteriophage so that it enters into the bacteria, integrates with the bacterial genome, but it is not able to divide and destroy the bacteria, right? So that means the virulent gene, the genes which make the virus responsible for destroying the host cell, those genes are removed, and that in that place we are putting the gene of interest. Okay, so in this case, and that's why we use the term disarmed. So we have removed the arms and ammunition which this virus has. Okay, so disarmed viruses, bacteria viruses are used to transfer gene of interest, in bacterial host. And in this, the usual bacteriophage which are used are lambda-phage virus, there is M13 virus, this is also a bacteriophage, these are there. Then there are these two, NCERT gives only these two vectors, but just a short list of the others as well. So we have some hybrid vectors where we have combined the plasmid and the bacterial DNA together. So those are called hybrid vectors. So in hybrid vectors, so here plasmid and bacteriophage DNA are combined. And this will of course act as a vector for bacteria only. Example here is, one is cosmid. So cosmid is basically plasmid plus lambda-phage DNA, and another one is phagemid. It has plasmid plus M13 DNA. Okay, these have more insert load capacity because bacteriophage can carry about within again 5-10 kb, but this can carry up to between 30 to 50 kb of DNA. So that way comparatively the insert load capacity is more. Then next one, next one we can mention is the shuttle vectors. Now shuttle vectors, just like shuttlecock, so they can transfer DNA or they can be used in a prokaryote as well as in a eukaryote. So they can move from prokaryote to eukaryote. Okay, so can operate between prokaryote and eukaryote host. Example here is YEP, that is yeast episomal plasmid. Now yeast also has a particular some yeast, Saccharomyces cerevisiae, this has a plasmid, that's a rare thing, and that has been utilized, and this can be used in a bacterial cell as well as in a this thing. Okay, so yeast episomal plasmid, okay, that is there. Then coming to some artificial chromosomes, as vectors. So in this we have bacterial artificial chromosome. So this must have come, this name, when you were doing human genome project. So for cloning human genes, because they are quite large, we have used BAC and YAC. Okay, so BAC, this is bacterial artificial chromosome. This one is prepared from F plasmid. So you see, up till now the plasmid that we mentioned, PBr322 and all, they were prepared from R plasmid, the plasmid in which antibiotic resistance gene is present. So that was R plasmid, but this one is prepared from F plasmid, which has fertility factor. Okay, so these are prepared from F plasmid, and these have more load capacity. So insert load, they can carry more. And the other one, that is YAC. YAC is of course yeast artificial chromosome. This one has the maximum load capacity. Okay, has maximum insert load capacity. Insert load capacity, that is, this question also has appeared, that is up to 1000 kb, or which can be 1 MB, that is mega base pairs. So these are, you see, some of the important vectors. Then but all these we were seeing only for unicellular organisms. Here of course, yeast we have moved on to eukaryote, but that too as a unicellular. So if we want to use a vector for a plant cell, and if you want to use a vector for an animal cell, so that I'll mention with the host itself, right? So the third category we can move on to hosts. So you see, for a genetic engineer, we had mentioned that first thing he would need is an enzyme to manipulate the DNA. Then that DNA has to be ligated to another DNA so that it can be sent inside the host cell. And host cell is providing the site or the environment where the recombinant DNA can make its copy as well as it can express itself. Okay, so hosts are the cells in which recombinant DNA is transferred and provide environment for replication and expression of DNA of interest, because along with this recombinant DNA, the DNA of interest has also come in them. Now these hosts can be bacterial cell, plant cell, animal cell, they all can be a host. So first of all, starting with bacteria as a host. Okay, so in this bacteria as the host, in which the most common bacteria used is E. coli. Once again, E. coli. There's a different strain for it, of course, K12 something. Now the thing is, that DNA, as your NCERT has mentioned, it's a hydrophilic molecule, and for any hydrophilic molecule to cross through the cell membrane, it requires channels, because if it is a water soluble substance, it cannot cross. Lipid soluble substances can cross, because any membrane is having lot of lipids in it, so one lipid can allow the other lipid to cross, but for a water soluble substance a channel is required. Now that protein channel is not present. It's not present, reason being this is not a usual thing that DNA can move in and out of cell, so there is no channel for it. So you have to move an elephant out of the room or maybe into the room, and you don't have a doorway for it. So it's something like that. So because DNA, you see, it's anyways the largest biopolymer and it is a hydrophilic molecule. So for this, so DNA is large biopolymer which is hydrophilic. So it cannot cross the cell membrane. Okay. So for this, we have to, host cell has to be made competent so that it can take up that DNA. Okay. So host cell has to be made competent to take up DNA. To take up DNA. Yes, there is a question, I suppose. Ashish, I told you we will be taking this up when I come to that point. Let me reach to animal host and then we will discuss it. Retrovirus, of course, is for animals only. Now, so host cell has to be made competent to take up DNA, right? And here one of the method which is given in your NCERT is the heat shock method. Okay, so heat shock method, where bacteria are incubated over ice, with divalent cations your NCERT has mentioned. So basically it is calcium salt. Of course it will dissociate. And now from this ice they are transferred to a solution which is at 42 degrees Celsius. Transferred to 42 degrees Celsius just for 2 minutes. Okay. And then immediately taken out and they are put back on ice. Then transferred to ice. Ice once again. Just maybe 2 minutes. Because more than that, bacteria will die. Still many of them die. So this kind of third degree that is given to the bacteria, then some of the bacteria they get transformed. Not all. Maybe out of 10,000 bacteria were there in your culture medium, out of that maybe just one gets transformed. So that's why you see why screening process becomes so important. Because we have to select out of the so many which are non-transformants, we have to select very few transformants. Okay, so in this what happens is that few bacteria get transformed, that is they take up the DNA. But the thing is what they are transformed, again we have to check whether the transformation is positive, that means they have taken the recombinant DNA, or that is negative, that is non-recombinant DNA, right? So that's a screening step has to be carried out. So this is one method. Then another is electroporation. So this is another method of transformation, making them competent. So in this electric pulse, for short duration electric pulse at very high voltage, but that is given. So some pores develop, transient pores, that is they are temporary pores develop, and through that the DNA will slip inside. Okay, so in this short pulses at very high voltage, very high voltage are given. So now what happens? Transient pores develop through which DNA enters. Through which DNA enters. Okay, this is one. And of course the third method. So one, as we see, NCERT has mentioned one of them. The another one I have mentioned with you, and third one of course using their natural enemies, that is bacteriophage. So using disarmed bacteriophage. So third method would be using disarmed bacteriophage. Okay. So these are various methods to transfer a gene of interest into the bacteria, which is the host here. Now taking up another one, that is plant cells as host. Okay, plant cells as host. Now in plant cells, if it is a dicot, then the commonest method is using Agrobacterium tumefaciens. So one method is using Agrobacterium tumefaciens. Now this bacteria, it's a soil bacteria. Okay, it's a soil bacteria, and it is considered as a natural genetic engineer. Natural genetic engineer, because what happens that if in the plant there is a injury, you see the outside epidermis of the plant, even that is providing it innate immunity, but if there is a cut on it, so from the soil, when the soil, because of the wind and all, it rises, so along with that this bacteria is also there, so that can easily enter through that wound. And when it enters, it has the ability, it has a particular plasmid in it, TI plasmid. So it can put a region of a TI plasmid, that's called tDNA. It can incorporate that tDNA anywhere at random in any of the plant chromosome at any point. Okay, so it has tDNA in its TI plasmid, which is integrated at random in any of the plant chromosome. In any of the plant chromosome. Okay. So here, once it integrates that, now this DNA is expressed. So do we have time? It's almost no. So that means rest we will have to, just 2 minutes are left. So from here onward we will take tomorrow, right? So further other viruses and all, what all we can use to transform a plant host. Then of course an animal host we will be discussing. And once this much part is done, the major work is done, and then we can just go through the steps involved in preparing recombinant DNA. So once you have this much base, everything would be easy. So let's wind up. Thank you students, it's almost time.